All right, ready to dive deep. Today, it's all about pharmaceuticals, but not the feel-good kind.
Ah, you mean the stuff we don't want in our meds.
Exactly, impurities. We've got a great prompt from a listener. Wants to unpack those ICH guidelines, Q3A through Q3E specifically.
Great area to explore. It's fascinating, really, how even a perfectly crafted drug can have, these tiny bits of unwanted stuff lurking in it.
Okay, so maybe not something you want to think about with every pill you swallow, but crucial nonetheless, right? For safety and
Oh,
effectiveness.
absolutely crucial. And that's precisely where these ICH guidelines come in. They're like our rule book, really, for making sure medications are safe and actually do what they're supposed to do.
Okay, so rulebook for impurities. Let's unpack that a bit, shall we?
Yeah.
You mentioned different types of impurities. What are we talking about here specifically?
Well, you can broadly break them down into a few categories: organic, inorganic, and then you've got those residual solvents. Each one, they pose their own risks and need their own approach to control.
Makes sense. I'm picturing, I don't know, some heavy metal leaching into a drug from the equipment. Is that a real thing that happens?
It is. Inorganic impurities, like those heavy metals, they often come from the manufacturing process itself. You've also got organic impurities, though, and those might be present in the starting materials. And then there's the whole degradation thing. A drug can actually break down over time.
So even more potential impurities to worry about. Makes you realize how complex this all is. How do they even find these things?
That's where it gets really interesting. We're talking super precise analytical techniques that can zero in and actually quantify these substances even when they're at super low levels.
So like finding a needle in a haystack, but on a molecular level.
Pretty much. One example is high-resolution mass spectrometry. Incredible tool. Lets us measure the mass of molecules with pinpoint accuracy so we can even spot tiny variations. Could be a sign of an impurity.
Super high-tech stuff. So we found an impurity. Now, how do we know if it's a problem? Like, is there a universal too much level? Or does it depend on the drug?
Definitely not one size fits all. The ICH guidelines provide a framework, but there are lots of factors. Toxicity of the impurity, for example, is huge. A highly toxic impurity is going to have a much lower acceptable limit.
Right, that makes sense. What about how the drug is taken, like, daily versus just once in a while? That must factor in, too.
Absolutely. Imagine a drug taken daily for a chronic condition versus, like you said, one for a short-term illness. The acceptable limit for, say, an impurity in that daily medication is likely going to be way lower, because of the longer exposure.
Okay, starting to see how this ties back to patient safety. It's a real balancing act.
Exactly. And that's where this concept of acceptable daily intake comes in, ADI. It's essentially how much of a specific substance can be ingested daily over a lifetime without causing harm. Plays a big role in setting limits.
So how do they calculate those ADIs and safety limits? Some magic formula?
Not magic, but definitely not guesswork either. Established methodologies, statistical models, the whole 9 yards to do a really thorough risk assessment.
Our listener actually mentioned statistical validation in their prompt. What's that mean here?
It's all about making those assessments as reliable as we can. Using statistical methods to analyze data, identify trends, really build confidence in those safety limits we're setting.
Sounds super rigorous.
Yeah.
Makes sense, though. Earlier, you mentioned drugs breaking down over time. How is that managed?
Stability testing is key. Imagine storing samples under different conditions, temperatures, humidity, light, and then we analyze them over time to see how they change. Gives us a real insight into shelf life and how to store them properly.
Sounds meticulous.
It is, but it impacts safety directly. A drug that goes bad quickly could be, dangerous. This helps us prevent that.
Okay, let's switch gears for a sec.
Oh.
Listener also asked about purge factors. What are those all about?
Purge factors, they're a measure of how effectively we remove impurities during manufacturing, like quantifying the cleaning power of each step, you could say. And of course, we calculate and validate those carefully to make sure the levels are safe in the end.
Interesting. So it's like each step is a filter, getting rid of more and more of those impurities.
Precisely. Now imagine we find a brand new impurity, something never seen before. Got to figure out quickly how risky it is.
That sounds tough.
It is. That's where QSAR comes in, quantitative structure-activity relationship.
QSR.
Yeah.
Okay, sounds a bit sci-fi. How's that work?
Might sound futuristic, but it's a real workhorse in toxicology today. We use computer models to predict how toxic a substance might be just based on its chemical structure.
So instead of a ton of experiments, you can use algorithms to get a first look.
Yep. It's super valuable when you're dealing with something brand new and don't have much data. Helps us prioritize.
Amazing. Technology is changing the game here.
It is, and it goes even further with all these new software tools being developed. They can help us define toxicological classes of impurities so much more efficiently.
This is fascinating. Feels like we're just getting started. It makes you realize how much work goes into ensuring that our meds are safe.
It's a hidden world, really, but one that affects all of us. That's what makes this deep dive so important. Welcome back to the deep dive. We've covered some fundamentals, but now let's get into those real-world complexities like you mentioned.
Exactly. Biopharmaceuticals, that's what really intrigues me. Seems like a whole other level when it comes to controlling impurities.
You're right, they do. Biopharmaceuticals, those drugs derived from living organisms, they tend to be much bigger, more complex molecules.
More complex than, say, the everyday pills most of us are used to.
Exactly. And that complexity makes them, well, more prone to a wider variety of impurities.
So we're not just talking about simple contamination anymore.
Right. With these biopharmaceuticals, we've got to consider things like product-related impurities, basically variations of the protein itself that pop up during manufacturing.
So the protein itself can have, what, little variations? How does that even happen?
Think of it like this. The cells making those proteins, they can make tiny mistakes. And those mistakes lead to, well, variations in the final product. They might have slightly different structures or some kind of modification.
So even a tiny difference could affect how the drug works. Wow. How do they even find those variations?
It's like detective work at a molecular level. We're talking advanced stuff. Mass spectrometry, for instance, can measure the mass of these molecules so precisely we can see even the smallest variations. And then there's chromatography, separates the components of the mixture, lets us zero in on each one.
Like a molecular jigsaw puzzle.
You got it.
Yeah.
And to help us solve that puzzle, we rely on guidelines like ICH Q5A, R2, and Q6B, specifically focused on those impurities in biopharmaceuticals.
These guidelines are like the gift that keeps on giving. Seems like they're behind every aspect of this process.
They are really, and they're always evolving too, just like our understanding of these drugs and the techniques we use to analyze them.
Makes sense. Okay, so we found those product-related impurities, but then there's still the stability issue, right? They can change over time, degrade, all that.
Absolutely. Stability is key, For any drug, really. It has to stay safe and effective throughout its shelf life.
Right. You wouldn't want a medication to, lose its potency or become unsafe just because it's been sitting around too long?
Exactly.
Yeah.
So that's where stability testing steps in. Remember those controlled storage conditions, different temperatures, humidity, light, that's a big part of it. We store the samples under those conditions and analyze them over time. See how they change. Helps us figure out how long they're good for and how to store them.
Sounds like a pretty involved process.
It is, but it's all about safety, right? Making sure that those medications are good to go when patients need them. And of course, there are guidelines for that too. ICH Q1A, R2 is a big one, lays out the framework. And there's also Q1F from the WHO that specifically focuses on active pharmaceutical ingredients.
So those guidelines keep everything consistent and rigorous, make sure those tests are done right.
Right. Ultimately, it's all having that scientific basis to know how long a drug is good for. And that's absolutely critical for safety, right?
Yeah.
Making sure that the medications people are taking are doing what they should be doing.
Now, what happens if a company has to make changes to the way they manufacture a drug? Like to make it more efficient or maybe address a problem?
Good question. That's where change management comes into play. Another key part of impurity control. Any change, even a small one, has the potential to affect the final product, including its impurity profile.
Because even a small tweak could introduce something new or change the levels, right? How do they make sure those changes don't mess things up?
That's where ICH Q5E comes in. This guideline tells us how to compare the drug substance or product before and after a change. The goal is to show that the final product is essentially the same, that it still meets those strict standards, even with those changes.
So like comparing before and after to see if anything went haywire?
Precisely. We might compare physical, chemical, even biological properties, maybe do more stability studies. Sometimes we even need new clinical trials, depending on the change.
Wow, that's really reassuring. Shows you how seriously they take this whole thing.
Absolutely. And that commitment to quality, it's at the heart of what we call a robust pharmaceutical quality system. That's what ICH Q10 is all about. It's not just about following the rules. It's about managing quality in every aspect, from design to manufacturing to distribution. The goal, making sure that product quality is consistent and patients are safe.
They're like this huge interconnected system all working together to keep patients safe.
That's a good way to put it. And managing impurities, well, it's a crucial part of that whole system.
Okay, we've covered a lot here. Biopharmaceuticals, stability testing, managing change. But I want to circle back to the why behind all this.
The why is always important.
Right. Why does all of this matter so much? What happens if we don't control impurities? What are the consequences?
That's a big question, and the answer is pretty straightforward. Failing to control those impurities can have serious consequences for patients.
It's easy to forget, right?
Yeah,
These impurities are tiny, we can't even see them, but they can have a real impact.
exactly. We've talked about how impurities can affect how well a drug works, maybe even making a condition worse. But in some cases, they can trigger serious, unexpected side effects.
So it could be anything from a mild reaction to something much worse.
Absolutely. That's why it's so important for drug companies and regulatory agencies to get this right. Every step, from the ingredients to the manufacturing, has to be carefully controlled to keep those harmful impurities out of the drug supply.
It all comes back to those ICH guidelines, right? Q3A through Q3E, making sure we can identify, classify, and control those impurities, and all those incredible analytical techniques that let us see what's really there.
Right. But it's more than just checking boxes. It's about really understanding the science and always striving to do better.
Always learning, always improving, all to protect patients.
That's the key. This deep dive, it's really pulled back the curtain on all the science, the regulations, the ethics that go into making pharmaceuticals. It's a complex system, but it all comes down to that commitment to patient well-being.
I'm with you. It's amazing, really, to think about how much we've learned about something that most people probably never think twice about. But as we've seen, it's incredibly important.
It is, and as technology keeps advancing and we learn more and more, the challenges and opportunities in this field are only going to get bigger.
That brings us to the next frontier. The future of impurity control. I'm excited to see what's coming next.
Me too. In the next segment, we'll dive into some of the most exciting developments, stuff that's shaping the future of safer, more effective medications.
Stay tuned. The best part of this deep dive is still ahead. Back again. We've covered a lot of ground, haven't we? Impurities, biopharmaceuticals, stability, those manufacturing changes.
It's been a journey, but like we've mentioned, this field is always moving forward. Time to look ahead, see what the future holds for impurity control.
I like that. Looking ahead, what's got you excited? Any breakthroughs on the horizon?
One thing that's really fascinating is how analytical technology is evolving. Scientists are creating these amazingly sensitive instruments, ones that can detect impurities at levels we couldn't even dream of before.
So pushing the limits of what we can see, what we can measure.
Exactly. High-resolution mass spectrometry, for example. We can measure the mass of molecules with incredible accuracy now, find even the tiniest traces of those unwanted substances.
It's like a super-powered microscope, revealing that hidden world of impurities. Amazing. How's that changing drug development?
Oh, it's a game changer. Think about it. If we can find potential impurities earlier, way earlier in the process, we can make better decisions.
Optimize that whole manufacturing process right from the start.
Precisely. We can adjust things to minimize impurities, reduce those risks before they even become a problem.
Like preventing the problem before it even exists. We talked about computational modeling, QSAR, predicting toxicity. What else is coming down the pike there?
Computational modeling, it's going way beyond just toxicity now. Scientists are building these really sophisticated models that can simulate the entire drug manufacturing process, from start to finish.
A virtual factory, in a way.
You could say that. And with these simulations, we can explore how all those different factors in manufacturing, those little variables, might affect how impurities form.
So countless experiments in a computer instead of in a real lab.
Exactly. We can optimize the process, minimize those impurities, all without the time and cost of traditional experiments. And as these models get more refined, more accurate, they're going to change how we approach impurity control, make it much more proactive.
Love that. Proactive, not reactive. Speaking of global things, what about those efforts to harmonize regulations? We touched on that earlier.
Right, global harmonization. Well, as the pharmaceutical industry becomes more global, we need to have consistent standards for drug quality and safety everywhere.
So, same rules of the game no matter where you are.
That's the idea. Harmonization efforts are all about getting regulatory agencies from different countries to work together, align those guidelines, those requirements, create a more unified system,
Makes a lot of sense. And ICH is playing a big role there.
Big role, yeah. They're bringing together experts from the industry and those regulatory agencies to make it happen.
Building bridges, making sure those standards are the same for everyone, everywhere.
Exactly. And it's not just about the agencies. More and more people are realizing that solving these impurity challenges, well, it takes everyone working together. Scientists, academia, industry, government.
All hands on deck.
Yep. That's how we'll stay ahead of the curve, you know?
Yeah.
Anticipate those new challenges as technology changes and we learn more about impurities.
It's pretty incredible when you think about it. All this amazing potential, all this progress. But at the end of the day, it's all about one thing.
Protecting patients.
Exactly. This deep dive, it's taken us from the tiniest molecules to the global stage. But that commitment to patient health, well, it's been the constant thread.
Couldn't agree more. This whole journey, it's about helping people understand, showing them the science, the regulations, the dedication that goes into every single medication.
I feel like I've gained a whole new appreciation for it.
That's what we hoped for. So the next time you pick up your medication, think about all the work, all the science that went into making it safe.
It's really remarkable. To wrap things up, let's recap some key points. We learned that impurities, well, they're just part of the game when it comes to pharmaceuticals. Controlling them is absolutely essential for safety and effectiveness.
We talked about all those guidelines, those incredibly precise analytical tools, how we identify those impurities, classify them, measure them, and how the challenges can change depending on the type of drug we're talking about.
Stability testing, that was a big one. Making sure medications stay good over time, and how companies handle those changes in manufacturing, make sure nothing goes wrong when they tweak things.
And of course, we looked into the future. All those exciting new technologies, those computer models that can predict problems before they happen.
It's a lot to take in. But the bottom line is this. Those pharmaceutical regulations, they might seem complicated, but they're there for a reason. To protect patients. To make sure they have access to the safest, most effective medications possible.
And that's what it's all about, isn't it?
Absolutely. Thanks for joining us on this deep dive into the world of pharmaceutical impurities. We hope you learned something new, maybe even got inspired to explore further on your own.
Keep asking questions. There's always more to learn.
Until next time.